the early years of your studies with aflatoxin, I guess you could think of those as preclinical studies, although maybe at the time you weren't thinking of it that way because you didn't realize that the pharmaceutical products that come out of this. What kind of preclinical research did you do?
Well, the type of work we've done with the aflatoxin has been bench work. A lot of what we call in vitro mechanistic studies to determine the mechanism of how this clay works to bind aflatoxin in the stomach and intestine. And we've looked at a variety of animals, especially with our colleagues at USDA, poultry and swine and lambs and dairy cows and caprine, dairy caprine.
Why did you use so many different species?
Well, because there's a big variation in how these are affected by aflatoxin, and in order to know you're dealing with a compound that's safe as you transition this into humans, you need to have a lot of species. We even use modeling procedures to model the toxin and model the clay and learn more about it, and even looked at thermodynamics. Now, what we did learn was that these were very selective based on our thermodynamic work, and that the clay, this particular clay we call Novosil.
How did clays get involved here? What caused you to start working with clays?
Well, we were looking at a lot of different sorbents, trying to find something, including antibodies, even a biological system that would bind this and activate it in the stomach and gastrointestinal tract. So the target was to develop an enterosorbent. But antibodies didn't work well, and other sorbents didn't work, and we found this clay.
These are some clays collected in Ghana, and these are edible clays that people used to eat in Ghana. And you can also find these pieces of clay. This is more like in a natural source. You find them with this shape, and the clays that we used for the study were placed in capsules like this, so people didn't know if they were taking the placebo or the clay treatment.
But a lot of clays are bad for you.
That's right. You have to be extremely careful because they come out of the ground, and just like plants and soil, can have a variety of priority metals that can be carcinogenic, arsenic, cadmium, mercury, lead, and a variety of others, as well as dioxins and furans.
All of that's bad stuff.
All of that's bad stuff, and so you have to, our particular clays that we looked at, we very carefully characterized for that, and we even have a process now where they're processed and they're refined to be sure that the levels of these toxins are less than what you would get in food. They're checked in animals for safety, and then they're checked to be sure that they don't interfere with nutrients, and Novosil doesn't. So we're very, very happy, that it binds aflatoxin very selectively.
Now, Novosil, apparently the name you've got for this pharmaceutical.
Correct.
It's in clinical trials now.
Correct.
What's the status of your clinical trials?
Well, the clinical trials that we're doing now, well, the first trial we did, in order to go to Africa because of the sensitivity when you're doing clinical trials, we ran the first study in the U.S. in Texas, and it was a phase 1 trial, which we call an adverse events trial, and we were looking at up to a high dose of 3 grams of this clay a day in a person, and to determine were there any adverse effects. Taken by mouth, capsule by mouth. To determine were there any adverse effects, based on health science center doctors, measuring clinical measures and blood measures and urine measures, and we found it was very efficacious and very safe. And that was the first study, and then recently we finished what we call a phase 2A study, and that was a 3-month intervention in Ghana.